Okay, it's time to start, and I hope you all watched the lecture video.
Could you please a bit clarify about decomposers? What are their functions exactly, maybe, or something?
Okay, nice. It's a nice question. So we have we have three types of organisms. First is the producer, so it's like plants. And second one is consumers. So they consume everything that is produced by plants, and decomposers. And decomposers, well, usually we say that it's fungi and bacteria. So they take organic molecules, complex ones, and they melt them, well, break them down, make smaller and simpler stuff from them. And in this way, they make them more accessible for consumers, okay? For example, there are a lot of molecules that humans' body cannot digest, something like cellulose, for example. And if there were not decomposers, we wouldn't be able to recover the sugars from the cellulose. And that's why we actually need these decomposers. So their role is essential, like we wouldn't be able to live without the decomposers. Is it clear?
Yeah, sure. I think so. They are like a part of a cycle, right?
For sure, yes, because a lot of debris are left by both producers and consumers. And both of these types cannot handle these debris, and only bacteria and fungi can do that for us. And we then regain these molecules from them.
Okay, got it.
There was like in the reproduction system, there is the thing, DNA is given from one body to another, from one organism to another organism. You said that there is also proteins and nucleic acids, and um, fats that are also important. Could you please clarify? Does proteins and does do proteins and fats are also important in conducting genes? That's kind of
Yes, it's called epigenetics. So everything that is encoded in the genome, so on the level of DNA and RNA, it is important to just carry the genes, the traits from parents to their children. And if we take epigenetics, this is everything that makes this genome open or closed, accessible for the proteins, for silencing, for actually translation, etc. So many proteins, they are involved in stopping the producing of the traits, like, you know, they just, how I can say that? They just stop making these traits visible for us. So they really change the phenotype. Do you see my screen?
Yes.
Okay, so just a second. So let's say you have the cell, and this is the nucleus, and we have DNA in it, okay? And this DNA carries the genes. The genes, every single gene encodes a trait, characteristic of the organism. In other words, it encodes the protein or RNA. Both of them are interactors, so they can have some function in the organism, okay? So these proteins are encoded in this DNA, which means we have some length of DNA, and many genes. For example, here we can have hemoglobin, let's say the IgG, etc., etc. Okay. So these traits, they are dependent on the DNA. But the thing is, not everything in the DNA, not all the genes in DNA, they are produced during the cell cycle. So one cell can actually produce hemoglobin, let's say like this, but do not produce the IgG. So how it chooses what to produce and what to not to produce. So we have another set of proteins, RNAs, some fats or sugars that are bound to this place, and they make it so they close the access to this part for this particular cell. Okay, so let's call this cell 1 and another cell 2. And for cell 2, we will not have these proteins on this site. So this part will be accessible, and this second cell will produce IgG.
May I ask, is this more about like message RNA and ribosomes that produce some type of proteins?
No, it's more about the process that kind of precedes the ribosomal translation. So before we switch to ribosomes, we need to produce the messenger RNA. And this messenger RNA is transcribed from this part of DNA, so from genes, okay?
Okay.
So again, what the cell produces, I mean, what type of genes it translates, depends on the proteins, mainly on proteins and RNA that actually closes or opens these genes. Okay, so that's why I said that proteins and RNAs and other compounds, they are playing a role in the gene regulation and reproduction.
May you please tell us more information about Archaea, that is in prokaryotic cells?
Yes, so I will tell you in just a second. So the Archaea, so you all know that we came from the same ancestor, and it's shown like something like this. So this is a phylogenetic tree, and here we have bacteria. Here we have Archaea. And here you have eukaryotes. So the thing is, this common ancestor is most probably was just a set of protein complexes and organelles, and at some point it formed the prokaryotes, and the prokaryotes don't have the nuclei, so they don't have isolated place for holding the DNA, so they usually have this circular DNA. And this circular DNA contains all the genes that carries these bacteria, okay? So this is for prokaryotes. For eukaryotes, it's easier to understand because we have a very distinct organelle inside our cell, which is surrounded by biomembrane. So it's a real border between the outside, which is cytoplasm, and the inside, which is the nucleoplasm. And all of our DNA is located here. So you might have heard about the chromosomes. So these chromosomes are in the nucleus and it is isolated from other sites, okay? The only time it is losing this isolation is when the cell divides and it needs to redistribute the copies of DNA. This time this biomembrane starts functioning and cell will form two places to pull this DNA on both sides. And then the cell divides with the equal amount of DNA on each side. This is how the eukaryotes work. And Archaea is something in between, but closer to eukaryotes. So prokaryotes. And here we have Archaea. So the thing is, it doesn't have the isolated nucleus, and it also is not floating inside the cytoplasm freely. It is bound to many proteins. So let's say this protein 1, and this protein 2, and protein 3. And it is bound, the DNA bound to these proteins. Just a second, so. So let's say this is DNA. And the thing about it, it's not isolated. It doesn't have this isolation membrane, and it's not floating. But these proteins, we can find them in the eukaryotic nucleus. Okay, which means Archaea might have had the common ancestor with us, with eukaryotes, but not with the bacteria. So we cannot say that they belong to bacteria, and we cannot say that it belongs to eukaryotes. Is it clear? Just a second. Okay, so the second question was about the DNA. So the DNA is deoxyribonucleic acid, and RNA, just a second, DNA and RNA. RNA is ribonucleic acid, and they differ by the number of oxygens inside their sugar. So if you remember the 5-carbonic sugar, so this is ribose. And on one of these sides, I'm not really sure which one right now. So we have OH, and in the DNA we have deoxyribose. It doesn't have this O, so it's more stable this way. So remember that DNA, because of this deoxidation, it's more stable, and RNA is more flexible. So one RNA with the same sequence of nucleotides, so let's say C, U, A, C, C, can form different tertial structures. So it might be like this, it might be linear, or many other forms. If you take the DNA with the same structure, for example, C, T, A, C, C, it will always form the same double-stranded chain, okay? Which means the RNA is not, how we say that, reliable for carrying the genetic information. So because of its flexibility, we cannot trust it anymore. Or when I say we, I mean nature. Nature couldn't trust it anymore, so it needed to come up with something new, something more stable that will be constant among the generations. And that's why we have DNA. And whenever we have information in the DNA, we can be sure that it will be carried in the same way to the offsprings. Is it clear?
Yeah, I'd like to ask about response to stimuli. I mean, how does it work? Why, for example, plants grow towards light? Just being curious.
Okay, so this all is controlled by chemistry. So whenever the plant starts growing, because of these billions of years of evolution, it already knows where the source of light is. And how it knows, just because there are sensing molecules that obtain the light, and they can control other molecules around them and try to kind of bend towards the source of life. That's why when you see the tree, on the southern part of it, just a second, again to the shared screen. Okay, let's say you have a tree. And this should be the north, the south, west, and east. So the thing is, just because we have more sun coming from south, okay, it means that on the southern part of the tree, we will have more leaves and more branches than on the northern part. We still have some of these leaves and branches here, but not as many as on this side. It's just because within these mechanisms that are responsible for development of the tree, we have molecules that sense this light. So just let's call them sensing molecule. So whenever it senses the light, it turns, let's say, this part of it towards the light. So it becomes like this. And because it is bound to other molecules, structural molecules, they will also turn towards light. And this will make this stem bend towards light. So instead of growing just in the right position, it will go a little bended towards light. Is it clear?
Yeah, sure. So,
Okay,
for example, when we see some plants like inclined, does it mean that they have some source of light from one direction?
Most probably, yes. So if you look at any plant, you will at least see how they kind of revert their leaves towards light. At least this modification happens in all plants. And if you have bending or inclining of the stem or the branches, it means that they are having strong attraction to the light.
Okay, thanks.
And almost the same happens in other organisms like unicellular organisms or multicellular organisms. So they have some sensors that understands the stimuli. For example, the needle. Whenever you poke yourself, you have sensing nerves that understand that there is a problem. So there is a danger coming from this needle. And most probably the response would be coming from your brain to these muscles to just reduce the distance between you and the needle. And this will be also the response to stimulus.
Excuse me. So the question that you asked here, like the first step of deductive method is hypothesis.
Yes,
Is it right?
it's the hypothesis based. So you kind of try to define the working model, and based on this hypothesis, you make predictions, and then you make experiments that can give you the glimpse of the predictions. Or, well, these experiments will give you the results that you will compare to the predictions, and then you say if the predictions are in order with these results, you will say that hypothesis is working, it is trustable, etc. Okay.
So, but if the first step of deductive method is hypothesis, the first step of inductive method is hypothesis 2,
Now it's observation.
observation.
Yes, so what is the difference between induction and deduction? So in the deductive method, you use huge, Rajan, could you please turn off the mic?
Okay.
Okay, so whenever you use the inductive method, you have huge amount of data. So you try to dig into it and try to define the rules that this data is created by. So you have rules that kind of drive this data. And then you make predictions based on these hypothetical rules, and you try to understand if it works or not. In the deductive method, it goes almost the other way around.
I will do. Later I will look at the book because I didn't really get it. Like I need to make more readings.
Okay, so let me try again, and then you can go to book. Okay, so in the inductive method, you have a huge amount of data. Then you try to kind of find the seeds of rules inside this data, define the rules that make this data happen. Okay, so something that creates this data, the rules that create this data. And then you try to understand if these rules can be trusted. And in the deductive method, you have the hypothesis, then you make an experiment based on this hypothesis, you obtain the results, you compare them to the predictions of this hypothesis, and then understand if you could prove or disprove this hypothesis, which means you don't use huge amounts of data. In the inductive reasoning, you start with a huge amount of data. This is the main difference between these two methods.
Okay, thank you.
And now you can go to textbook. Well, usually plants do like the sunlight. The thing is, some of them might like a small part of it, which means like the shadows, etc. That's why they usually don't grow too tall. I mean, the small trees, they are very short, and they stay under the coverage of bigger plants. That's how they regulate these sunlight beams.
Mr. Radmir, I want to ask a question.
Yes.
So when you watched the video, there was information that metabolism is all the chemical reactions happening inside the body. Yeah? So can we connect homeostasis, reproduction, and other this kind of processes to the metabolism?
Yes, they are part of metabolism, because to make sure that you have a stable homeostasis, you will need to perform many chemical reactions, like equalizing the concentrations or making concentration of one element less than inside than outside, and it needs energy and energy is usually spent for chemical reactions. So these transformations of energy is metabolism. So it's kind of driving the homeostasis. So this data is collected through observation. Yes, this data is collected through the observation. I guess this question is related to the question about deductive and inductive methods. So the main difference between the data collection in inductive method, we just observe. We don't perform any kind of movements. We don't perform any experiments. We just observe. And when we collect the data, we try to understand how it's created and how it works. In the deductive method, we usually collect the data from the experiment, or we collect the data from the natural phenomenon, which anyway, this data will not be as huge as in inductive method.
Can you clarify that I might be unsure about taxonomy levels of classification? I didn't understand. What is the important main points about taxonomy?
Okay, so actually everything about taxonomy is important because you will, just a second. This is the passport of the species. So this is the whole kind of name of the species. So you will need to understand every single bit of it to know how to describe the species. For now, we are using just genus plus species. But usually, even if you go, let's say, to Wikipedia and try to find Homo sapiens, you will see that they actually say all the traits like species, genus, family, order, class, phylum, kingdom, and then domain, because this is science and you will need to be, how to say that, very precise in explaining something. Sometimes there might be cases when two very close stuff, very close animals, for example, they look similar, but they are from different, just a second, from different orders, for example, from different families.
What about cladistics?
Okay, so the cladistics is more about defining the relationships between the species. So that's how we define which family to relate these species to. And this cladistics as a field of science uses molecular biology methods. It uses the genetical comparisons, etc. So according to these cladistics, you can build this phylogenetic tree. For most of the time, the main task of cladistic was to define the common ancestors of the species.
Excuse me, can I ask the last question? So if looking at the taxonomy of the person, of people, where are we different from primates, for example? You gave the example, like we have...
We are not different from primates, we are primates.
So we are primates, okay.
Yes.
Could you
So
please
there are
clarify?
many, yeah, so it's like rodents. For example, you have many species of rodents, but not all of them can evolve into each other. The same happens in primates. Not all primates could evolve into humans. And we are from the same order, from the order of primates, but our families are different. So some families will never give, even if they start evolving very intensively, they will not have the traits that make humans humans. So let's say the volume of the brain, this is a huge characteristics. And even the muscles on your palm. Not all primates have the same set of muscles. They might have analogs, but the one that makes us take stuff like this, I mean, making this movement, is not present in all primates. It's not even present in all hominids. Okay.
Excuse me, can I ask one question?
Of course.
In the lecture video, you had a lecture slide where tattoos were shown. Can you clarify
What?
this slide about phylogenetic tree? In the lectures, you showed the picture of tattoos and
Okay.
mentioned some of the ancestors and other examples. Can you clarify this?
Okay, so
Yes,
the thing
this
is,
one.
here you can see like right in the middle, you can see the start of the tree, I mean the root of the tree. And this is the most common ancestor of prokaryotes, archaea, and eukaryotes. So starting from here, we start dividing into eukaryotes and prokaryotes, for example. So this one is not very precise one because you can see here connected parts, and you will not see the same in phylogenetic tree, but the artist saw it like this, and at least they use science in it. But for this example, you can see, for example, the common ancestor for everyone here will be on this side, on this shoulder, and here, because everything starts from them. Do you see it? So it's like, you know, branching. So you have the single stem, the main one, and then you have small branches that will have branches themselves. Okay? Same here, for example. So you have a single root, and everything comes from this root. So if you try to understand, for example, this guy here, you can go along this line. So you go back to the common ancestor, and finally you come right here, okay? And same happens here. So this is the single root that actually gives the eukaryotes and prokaryotes. It's actually a very interesting topic because when we find the common ancestor for prokaryotes and eukaryotes, it will be a huge discovery, because understanding the processes that happened in those small bodies will tell us how the dead chemistry became live chemistry. So it's a very interesting topic.